All right, the next thing we're going to talk about is called electroencephalography. Does someone have a guess for what this means? Anybody? Yeah, go ahead.
Finding a way to map out the electrical impulses in the brain.
Yeah, perfect. That was a perfect explanation of what it is. You're mapping out the electrical impulses in the brain, and so it's all in the word. Electro means electricity, encephalo means the brain, and then graphy is measuring stuff. So that was a really good answer. All right, so earlier we measured electrical activity from sensory neurons in a leg, and that was pretty easy to do. We just poked the leg, put an electrode in there, and we could measure the signals. But for the brain, that's more complicated. Can someone think of some reasons why measuring electricity from the brain would be more of a challenge? Yeah.
You run the risk of accidentally messing something up.
Yeah, certainly. If you poke something in your brain, then you could damage those cells, and then you could end up with some serious problems. Yeah. What else?
There are a lot of activities going on.
Yeah, that's right. So the brain is pretty big, and it's really chock full of neurons, and all those neurons do different things. So figuring out where activity is happening, what that activity means, what process is going on there, it's very complicated. So these are the two challenges. You have a lot of area that you need to record from, and if you poke stuff in there, you can damage it. So there are some different ways that researchers try to measure brain activity, and they have, given these challenges, they have benefits and drawbacks. So the first one is called an fMRI or a PET scan. So these scanning methods look at very broad regions of the brain, and they can tell you if activity is happening in that region, but it can't tell you exactly what is happening or what individual neurons are doing. But the benefit of this is that you don't have to poke anything in there. You don't have to open up the brain or anything like that. But the drawback is that it's kind of imprecise. Another way of doing this is the EEG, and this EEG stands for electroencephalography. So that's specifically what we're talking about, or what we'll be doing later. And this method is similar to the scans in that it measures activity from bigger regions of the brain, because you're actually measuring from outside the skull. So again, this is not invasive, and you're not in danger of damaging anything, but you only get information from chunks of brain. The benefit of EEG over scanning is that it's a more direct measurement of electricity. So you're not just getting this region is active, but you're actually getting what type, like electrical signals from that region. And then the most precise method would be an electric implant. So that would be like the chip that you saw in the video earlier. And this would be implanted directly into the brain. This gives you very precise information about individual neurons or small groups of neurons and the electrical signals that they're sending. But it's invasive. So you run the risk of damaging something, and this would require very difficult surgery, opening the skull, accessing the brain. So in a human, this really wouldn't be done unless the human is undergoing surgery for some other reason, or they have a really serious problem. So yeah. So if you guys had a choice of which way you want to look at your brain activity in class today, which one do you want to do? EEG. I hear you guys pretty unanimously saying EEG, and you're in luck because that's what we're going to do. So when you're doing EEGs, as I mentioned, you have these electrodes on your skull, so that electrode will pick up activity from the whole chunk of brain that's below that spot. So that means that you have many thousands of neurons that this one electrode is listening to. And that makes things a little bit complicated. So pick up your piece of paper. We're going to do a little demonstration of what it might be like for the electrode that's listening to many neurons talking at the same time. All right, for this activity, you have 3 words on your sheet. And just look at the first one, the one in the first box. And on the count of 3, we will all say this word at the same time. You ready? Okay, 1, or after the count of 3, not 3. 1, 2, 3.
Stop.
Okay, listening to the room, could anyone pick out a message? Do you think so? No, you seem pretty unsure. Okay, let's move on to the second one. Ready? 1, 2, 3. Light waves. Did anyone think they could pick out a message over all the noise? Something that stood out to you? Yeah?
I heard her say lateral interface.
Okay.
Yeah, interface, right?
Okay, but it was kind of difficult to hear it because there's a lot of different stuff going on. All right, let's try the last word on the count of 3. 1, 2, 3. Visual cortex. Did anyone hear anything that stood out more than something
You
else?
said the same thing I said it.
Yeah, what did you say?
Visual cortex.
Visual cortex. Did anyone else hear visual cortex over the noise? Great, a lot of people. Yeah, so that demonstrates what it's like for the electrode, listening into many neurons talking at the same time. And another way to think of this is as a big stadium or a classroom full of people, where if you're all saying different words, like we did for the first round, you can't tell what's going on. It's just a lot of people or neurons saying different things, and it just all the signals cancel out basically, and you can't tell what's going on. But if more people are saying the same thing, so what in the third round, when all of us or a lot of us said visual cortex, you could pick that out. Or in a stadium, if all the people are singing the national anthem, they're all doing the same thing, the activity becomes synchronous, and then the electrode listening from outside can tell what's going on. So all these individual neurons are connected to each other through synapses, and this creates what's called a synaptic network. So when all these all the neurons are connected together, we get a synaptic network, and with all these different neurons connected to each other, this activity, whether they're doing different things or becoming more synchronous, it looks like waves. So that's a feature of networks, that their activity, if you look at the sum of it, will look wave-like. And you've probably heard before about brain waves, and there are different types of brain waves. And the shape of the brain wave is determined by how synchronous or asynchronous the activity of the network is. And interestingly, the smaller your wave, the more awake you are, and the more conscious activity you're doing. And the slower your wave, the more unconscious you are. So the very smallest wave at the top is called the beta wave, and that means that you are awake and maybe doing some problem solving. Whereas the alpha wave means you're still awake but probably at rest, maybe meditating, and your waves have slowed down because your neurons have become more synchronous. Then a theta wave means you're becoming unconscious, maybe at the beginning of sleep, and then a delta wave is when you're in very deep sleep and you're not dreaming, so there's really very little activity going on in the brain. It has just all synced up together. So since we all said visual cortex, how does this pertain to the visual cortex? If you look at a very complicated visual scene while you're awake, then all the neurons in your visual cortex will be doing different things, which means their activity is what? It will be a beta wave, yes, because the activity is asynchronous. So all the neurons are doing very many different things, all the signals will cancel out and will look like a very small, fast wave. On the other hand, if your eyes are closed, you're still awake, but your eyes are closed and all your neurons are seeing the same thing. Now the activity of the neurons is synchronous. So if the activity is more synchronous, the wave will be slower. So if you measured this, you would see not a beta wave, but a slower alpha wave. Right? So we will quickly try this on a person and see if that's what we see. So who wants to volunteer? And it will actually be easier to do on a person with short hair. Perfect.